The next stage of photosynthesis is the Calvin cycle. The Calvin cycle uses ATP and NADPH to convert carbon dioxide to sugar in an anabolic pathway. The cycle happens 3 times, fixing 3 carbon dioxide molecules to make 1 molecule of G3P, which is the starting material for producing glucose. Carbon fixation incorporates each carbon dioxide molecule one at a time by attaching it to a 5-carbon sugar called ribulose biphosphate. We will refer to it as RUBP. RuBP carboxylase, also known as Rubisco, catalyzes this first step, producing a 6-carbon intermediate that is so unstable it immediately splits into 2 molecules of 3-carbon 3-phosphoglycerate for each carbon dioxide fixed. Reduction starts when each molecule of 3-phosphoglycerate receives a phosphate group from ATP, forming 2,1,3 biphosphoglycerate. Using all 3 CO2, this step would use 6 ATP, leaving behind 6 ADP. These then receive 2 electrons from NADPH, reducing their carboxyl groups to aldehyde groups, which store more potential energy. Next, the phosphate groups are lost, so 2 G3P are formed. G3P is also called glyceraldehyde 3-phosphate. For every 3 molecules of CO2, 6 molecules of G3P are formed, but only 1 molecule of G3P exits the cycle to be used for cell activity. The other 5 molecules are recycled to generate 3 molecules of RuBP. RuBP is generated by spending 3 ATP, each losing a phosphate group, to make 3 RuBP from 5 G3P. The cycle then can restart.